day, this is the Professor, CBMD PhD, and today's Monday, August 7th, 2023. It is 4:33 and 40 seconds, AM Eastern Standard Time, and today's research, immune regulators. So let's jump to it here. A study finds a surprising new role for a major immune regulator. In addition to turning on genes involved in cell defense, the STING protein also acts as an ion channel, allowing it to control a wide variety of immune responses. Date August 3rd, 2023. Source: Massachusetts Institute of Technology. The immune regulatory protein STING has a previously unknown function, acting as an ion channel that allows protons to leak into cells. This discovery makes it the first human immune sensor that can translate danger signals into ion flow. As STING is primarily on the lookout for DNA, which can indicate either a foreign invader, such as a virus or damage to the host tissue or a cell. When STING detects a danger signal, it can turn on at least 3 different pathways, one leading to interferon production, one to non-canonical autophagy, involving recycling cell components and clearing pathogens, and a third to formation of the inflammation, a complex of proteins that activates inflammatory response. The mechanism by which STING stimulates interferon production is well characterized, but it has not been understood how it activates the other two processes. The scientific investigators and microbiologists has discovered how STING activates those two pathways. They found that STING has a surprising and previously unknown function. It can act as an ion channel that allows protons to leak out of an organelle known as a Golgi body. This makes it the first human immune sensor that can translate danger signals into ion flow. Arriving at this new idea, that STING is a proton channel, require connecting prior findings by other laboratories that either STING or proton flux could activate the inflammation and non-canonical autophagy, which led them to hypothesize that STING initiates or mediates proton flux to trigger both downstream processes. And because of its importance to host immunity, there is a great interest in developing drugs that can activate or suppress STING activity, and the discovery of STING ion channel activity will provide new ways to think about designing therapeutics to modulate STING. As STING, short for stimulator of interferon genes, is considered one of the major factors that triggers the immune response in the context of infection, autoimmunity, and cancer. And drugs that activate STING have been developed and tested in clinical trials as cancer immunotherapy drugs that would help stimulate the immune system to destroy tumors. As STING is a protein that can span membranes, and it is usually found embedded in the membranes of an organelle called the endoplasmic reticulum. Once it detects DNA, it relocates to the Golgi body, where it begins to activate proteins that turn on genes required for interferon production. And people know pretty well how STING induces interferon, but how STING induces autophagy and inflammation formation has been an open debate in the field for the last 10 years. As previous research has shown that both autophagy and formation of inflammation, large protein complexes that stimulate inflammation, can be provoked by protons leaking from cell organelles, which makes the inside of the cell more acidic. Because of that, the researchers wondered if STING might somehow induce proton leakage. And to explore this possibility, the researchers labeled the Golgi with a protein that fluoresces when the pH goes up. When they treated the cells with a molecule that activates STING, the Golgi became less acidic, meaning that it was losing protons. A genetic screen minimized the possibility of another ion channel controlling this ion flow, so the researchers hypothesized that STING itself was acting as a proton channel. In addition to its biological significance, this study is a notable example of the maturing functional genomics field, where pooled screening data are sufficiently reliable to set the direction of focused investigation, even from negative results, as was the case here. After running the structure of the STING protein through a computer model that can predict whether a given protein structure might contain a pore. The researchers found that the sting protein is predicted to contain a region that resembles a pore. And serendipitously, last year a company that was seeking sting agonists, molecules that activate sting, found a new molecule that was later shown by an academic group to bind in this precise location. The researchers hypothesized that the agonist, known as C53, blocks a putative pore. When C53 was added to cells, proteins did not leak out to the Golgi and downstream pathways, activating autophagy and inflammation formation were not turned on, even if sting was activated through other means. However, interferon activation, which goes through a different pathway, still occurred. And for the first time, they were able to decouple these downstream processes where they can activate interferon with C53 but inhibit those other two pathways leading to autophagy and inflammation formation. In the context of inflammatory diseases, where sting is overactivated, they can now start asking which of those molecular mechanisms is most important and contributes the most to the phenotype that they are seeing. In future studies, the researchers hope to use C53 to determine the relative importance of these three pathways and try to figure out which ones would be most useful to stimulate or to block to treat a variety of diseases. The U.S. Food and Drug Administration has not approved any sting agonists thus far, although multiple clinical trials are currently underway. One potential reason that other sting agonists have not made it beyond clinical trials is that the treatment can result in unwanted cell death mediated by sting. As drugs that stimulate interferon production but not cell death or other inflammatory pathways can offer a way to overcome that obstacle, according to scientific investigators and microbiologists. The researchers hope to explore whether sting might play a role in influencing the behavior of other cellular activities known to be controlled by ion channels. Now that we know that sting is an ion channel, we can propose other effects that we think occur based on this technology that sting does transport protons. And this is a professor of microbiology, bioinformatics, pathophysiology, and nature of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.